Hello everyone, welcome back to our channel. So today we are going to talk about the agrobacterium-mediated gene transfer. As we have seen in the previous video of the Vir genes in the TI plasmid, so these Vir genes are actually responsible for integration and transfer of tDNA into the genome of plant cells. Now here we will see we have the TI plasmid and we have the tDNA. So this tDNA is transferred as a single-stranded tDNA into the plant genome, and this is integrated into the plant genome. We will see here the Agrobacterium tumefaciens when it infects the plant cell, how exactly it infects the plant cell. As plant suffers the wound, and in response to this, it releases the acetylcysteine, and the agrobacterium is attracted to the plant. Now here we have different types of proteins like the Vir A and Vir G proteins. So Vir A and Vir G, they act as the in taking the signal like the acetylcysteine, and Vir A gets phosphorylated at the histidine residue, and Vir G gets phosphorylated at the aspartate residues. And in this way, this Vir G protein, this then in the form of a dimer, activates the other Vir operon genes. And here we have the tDNA region that is flanked by the left border sequence and right border sequence. And at the right border sequence, Vir D1 and Vir D2 proteins, they get bind. And in this way, this Vir D1, this acts as a topoisomerase, and Vir D2, this will act as an endonuclease, and it will cut out that single-stranded tDNA. And polymerase initiates the synthesis of tDNA that is being replaced, and then Vir E proteins get bound to this single-stranded tDNA, and this is the looping tDNA, and here we get bound the Vir E proteins, and nearly 600 Vir E proteins, they bind to the single-stranded tDNA to be transferred. Now this tDNA bound with the different types of proteins, the Vir E proteins and Vir D2 protein, this is ready now to be incorporated into the plant. This will be incorporated into the plant genome now. So zooming out this Vir D2 bound protein, this tDNA, now Vir B will initiate the channel formation. So it forms a tube for the passage of the tDNA, and Vir B11 will join the Vir B in formation of the channel. And also the Vir D4 proteins, they will get bound to the Vir B and Vir B11 so that the tDNA is transferred into the plant genome. Now this this tDNA will get inside the plant cell guided by the Vir D2, and channel forming proteins, they get disintegrated, and now this tDNA is actually incorporated into the plant genome by the help of the homologous recombination. And in this way, the crown gall tumors, they appear on the plant. Bacterium tumefaciens, as this is the natural genetic engineer, different molecules have to be released at the wound site so that it can attract the agrobacterium, and in this way the agrobacterium transfers its tDNA into the plant genome. So this is the natural genetic engineer why the crown gall disease happens. As this agrobacterium DNA transfer system, this is actually a powerful vector system used for plant transformation. Any genes put in the tDNA region, they get transferred to the plant genome. The DNA is inherently stable once in the plant genome. Since neither the border nor the virulence genes are transferred, Agrobacterium has been proved to be incredibly useful for the transfer and insertion of genes into plants. But its scope is limited to the plants that can be infected by the bacteria. And monocots are not the natural hosts of Agrobacterium tumefaciens, and hence most monocot plants are generally resistant to the Agrobacterium infection. And it has been actually argued that Agrobacterium infection of monocots is inefficient because wounded monocot tissues, they do not produce the phenolics such as acetylcysteine at the sufficient levels to induce Vir gene expression. It provided possible to induce tumors in certain monocot species such as asparagus and yam, and in recent years amazing progress has been made in the transformation of cereals using agrobacterium. And the first species to be transferred was the rice. And the breakthrough in cereal transformation using agrobacterium reflected the recognition of a number of key factors required for efficient infection and gene transfer to monocots.